Chapter 11 Efferent Division Autonomic and Somatic Motor Control. First, we'll dive into the autonomic division, which manages involuntary functions like heartbeat, digestion, and breathing, keeping us alive without conscious effort. Next, we'll explore the Somatic Motor Division, which enables voluntary movements, like lifting your arm or kicking a ball, by controlling skeletal muscles. By the end of this chapter, you'll understand how these systems work together to keep you moving and functioning smoothly. Let's get started. In this section, we'll explore the efferent division of the peripheral nervous system, which plays a crucial role in carrying commands from the central nervous system, or CNS, to the muscles and glands of the body. The efferent division has two main subdivisions. First, the somatic motor neurons, which control skeletal muscles. These neurons allow us to perform voluntary movements, such as walking, writing, or waving hello. Second, the autonomic neurons, responsible for involuntary actions. These neurons control smooth muscle, cardiac muscle, many glands, and some adipose tissue, keeping vital functions like heart rate and digestion running automatically. Now, let's take a closer look at the autonomic division of the nervous system. The autonomic division has two key subdivisions. First is the sympathetic branch, often described as the fight or flight system. This branch prepares the body for stressful or emergency situations, increasing heart rate, dilating pupils, and redirecting blood flow to muscles for quick action. Second is the parasympathetic branch, known for its rest, digest, defecate, and urinate functions. It helps the body relax, conserve energy, and carry out essential maintenance activities like digestion. These two branches are anatomically distinguishable, but are best understood by how active they are in different situations. While the sympathetic branch kicks in during emergencies, the parasympathetic branch dominates during calm, restful moments. This illustration shows how homeostasis, or the body's stable internal environment, is maintained through a dynamic balance between the autonomic branches. On the left side, you'll notice the parasympathetic branch, responsible for the rest and digest functions. Here, parasympathetic activity dominates, allowing the body to relax, slow down, and carry out everyday processes like digestion and recovery. On the right side, the sympathetic branch represents the fight or flight response. In stressful situations, like encountering a snake, sympathetic activity takes over, heightening alertness and preparing the body for quick action. These two branches work like a seesaw. When one side is active, the other is less so. This constant adjustment ensures your body responds appropriately, whether you're relaxing or facing a threat. Striking this balance is essential for maintaining homeostasis and overall well-being. Autonomic, or visceral, reflexes play a crucial role in maintaining homeostasis. These reflexes begin when the hypothalamus receives sensory input from various sources, such as the limbic system, sensory tracts, and cranial nerves. This information helps the body monitor both internal and external environments. The output from the hypothalamus is directed to several key areas. The medulla, particularly the reticular formation nuclei, regulates vital functions like cardiac activity, blood pressure, and breathing. The vagus nerve, or cranial nerve 10, controls parasympathetic responses, while the sympathetic lateral horns located in the spinal cord segments T1 to L2 coordinate sympathetic functions. In addition to these neural pathways, the hypothalamus activates the endocrine system. For example, it releases antidiuretic hormone, or ADH, when there is an increase in extracellular fluid osmolarity, helping the body retain water. Behavioral responses, like increased drinking when dehydrated, also stem from hypothalamic signals. The bottom line is that autonomic reflexes integrate sensory input with hypothalamic commands to coordinate efferent outputs that preserve homeostasis. Interestingly, not all autonomic reflexes involve the brain. Some, like spinal reflexes responsible for micturition and defecation, occur directly through the spinal cord, allowing for faster responses. This figure highlights the autonomic control centers of the brain, which play a critical role in regulating involuntary functions essential for maintaining homeostasis. At the top, the hypothalamus is the primary control center. It monitors and regulates key processes such as temperature control, water balance, and eating behavior. By receiving sensory information, the hypothalamus adjusts body functions to maintain stability. Below the hypothalamus, the pons helps regulate urinary bladder control and houses the secondary respiratory center, working alongside other brain regions to control breathing patterns. Further down, the medulla manages vital autonomic functions like blood pressure control and acts as the primary respiratory center. Its ability to adjust heart rate and breathing ensures immediate responses to changes in the body's environment. Together, these control centers form a network that continuously monitors the body's needs and adjusts its functions, ensuring survival without requiring conscious effort. A key feature of the autonomic division is antagonistic control, where most internal organs are regulated by both the parasympathetic and sympathetic branches. Typically, one branch is excitatory, stimulating organ activity, while the other is inhibitory, reducing it. This balance allows the body to adapt quickly to changing needs. However, there are some exceptions to this dual control. For instance, sweat glands and the smooth muscles in most blood vessels receive input only from the sympathetic nervous system. This is known as tonic control, where the level of sympathetic stimulation determines the degree of response rather than switching between opposing inputs. Interestingly, there are situations of cooperative control where both divisions work together toward a common goal. A prime example is male arousal, where different tissues are influenced by both systems to coordinate a complete physiological response. It's important to note that the neurotransmitter receptor on the target tissue often determines how the tissue responds, highlighting how the same chemical signal can have varying effects depending on the receptor involved. Autonomic pathways are characterized by having two efferent neurons arranged in series. These two neurons work together to transmit signals from the central nervous system to the target tissue. The first neuron in this chain is the preganglionic neuron. Its cell body is located within the central nervous system. It projects its axon out of the CNS to an autonomic ganglion, which is located outside the CNS. Here, it forms a synapse with the next neuron in the pathway, known as the postganglionic neuron. The postganglionic neuron is the second neuron in the series. Its cell body is located within the autonomic ganglion. From there, it extends its axon toward the target tissue, where it synapses and delivers the final command to the organ or tissue being regulated. A ganglion is essentially a cluster of neuronal cell bodies located outside the CNS. While ganglia typically contain neurons that connect the CNS to peripheral tissues, they may also contain neurons that are completely contained within the ganglion itself. Together, these two neurons ensure that signals from the brain or spinal cord can efficiently reach organs throughout the body, enabling the autonomic nervous system to regulate involuntary functions effectively. This figure illustrates the structure of autonomic pathways, which consist of two neurons that work in series to transmit signals from the central nervous system to the target tissue. The process begins with the preganglionic neuron, which originates in the central nervous system. Its axon extends outward, traveling to an autonomic ganglion, a cluster of neuronal cell bodies located outside the CNS. Within the autonomic ganglion, the preganglionic neuron forms a synapse with the postganglionic neuron. From this point, the postganglionic neuron carries the signal away from the ganglion and projects its axon to the target tissue, where it delivers the final command that triggers a physiological response. This two-neuron chain ensures efficient communication between the CNS and various organs, allowing the autonomic nervous system to regulate vital involuntary functions like heart rate, digestion, and respiration. The sympathetic and parasympathetic branches of the autonomic nervous system originate from different regions of the central nervous system, allowing them to carry out distinct functions. The sympathetic branch arises from the thoracic and lumbar regions of the spinal cord, specifically from segments T1 to L2. Its nerve fibers connect to a series of interconnected sympathetic chain ganglia, which form two chains running along either side of the vertebral column. This arrangement enables the sympathetic system to rapidly coordinate widespread body responses, such as during the fight or flight reaction. In contrast, the parasympathetic branch has a craniosacral origin. It begins in the brainstem and exits the brain through various cranial nerves. Additionally, it emerges from the sacral spinal cord levels S2, S3, and S4. Unlike the sympathetic system, parasympathetic ganglia are located on or near the target organs, allowing for more localized and specific control, often associated with rest and digest activities. One of the most important components of the parasympathetic system is the vagus nerve. This nerve carries approximately 75% of all parasympathetic fibers. It transmits sensory information from internal organs back to the brain and sends motor signals to thoracic and abdominal organs, playing a crucial role in regulating functions such as heart rate, digestion, and respiratory rate. This figure illustrates the sympathetic division of the autonomic nervous system and its widespread influence on various organs throughout the body. The sympathetic pathway begins in the hypothalamus and reticular formation, with descending signals traveling through the pons, medulla, and down the spinal cord from segments T1 to L2. The sympathetic chain, located on either side of the vertebral column, enables the rapid distribution of nerve signals to different target organs. This system is essential for initiating the fight or flight response, preparing the body for action during stressful situations. When activated, the sympathetic nervous system triggers several physiological changes. It causes the pupils to dilate, enhancing vision, and stimulates the salivary glands to secrete mucus and enzymes. In the cardiovascular system, it increases heart rate and contractility to improve blood circulation. The airways relax to allow for easier breathing. In the digestive system, sympathetic activation inhibits digestion, decreases enzyme and insulin secretion, and reduces gastrointestinal motility, redirecting energy away from non-essential functions. The adrenal medulla is stimulated to release catecholamines, including adrenaline, which further amplifies the fight or flight response. In the urinary system, sympathetic stimulation relaxes the bladder and contracts the sphincters, delaying urination. It also induces ejaculation in males and stimulates uterine contractions in females. This integrated response ensures that the body is prepared to face immediate challenges, enhancing survival during times of physical or emotional stress. This figure illustrates the parasympathetic division of the autonomic nervous system and its calming, restorative effects on the body. The parasympathetic pathway begins in the hypothalamus and reticular formation, with signals traveling through the pons, medulla, and extending along the cranial nerves and sacral spinal cord segments, particularly S2 to S4. The vagus nerve, shown prominently in this diagram, plays a central role by carrying signals to most thoracic and abdominal organs. This nerve slows the heart rate, encourages airway constriction, and stimulates digestive functions. In the eyes, parasympathetic stimulation causes pupil constriction, improving near vision. It activates the salivary glands, promoting watery secretion that aids digestion. Within the gastrointestinal tract, it increases bile secretion from the liver, stimulates stomach activity, and enhances motility and secretion in the intestines, facilitating nutrient absorption. The pancreas responds by releasing enzymes that further break down food, while the adrenal medulla is not directly innervated by the parasympathetic system, highlighting a distinction between the two autonomic branches. In the urinary system, the parasympathetic nerves trigger the release of urine by stimulating bladder contraction. In the reproductive system, they induce erection in males and promote engorgement and secretion in female reproductive organs, supporting sexual arousal. Overall, the parasympathetic system's primary goal is to conserve energy, enhance digestion, and restore the body to a state of rest and recovery after periods of stress or exertion. This table provides a side-by-side comparison of the sympathetic and parasympathetic divisions of the autonomic nervous system, highlighting their key differences. The origin in the central nervous system differs between the two branches. The sympathetic division arises from the thoracic and lumbar segments of the spinal cord, specifically from T1 to L2. In contrast, the parasympathetic division originates in the brainstem and the sacral segments of the spinal cord, particularly from S2 to S4. When examining the ganglion location, sympathetic ganglia are positioned close to the spinal cord. This arrangement allows for quick signal transmission and coordinated responses throughout the body. In comparison, parasympathetic ganglia are found on or near the target organs, providing more localized and precise control. The pathways of these systems also differ. In the sympathetic division, there are short preganglionic neurons and long postganglionic neurons, allowing for widespread distribution of signals. Conversely, the parasympathetic division features long preganglionic neurons that extend directly toward the target area, followed by short postganglionic neurons, enabling focused and specific responses. Together, these structural differences reflect the unique roles of each division. The sympathetic system prepares the body for rapid action during stress, while the parasympathetic system promotes rest, recovery, and energy conservation. The autonomic nervous system uses a variety of chemical signals to communicate between neurons and target tissues. These chemical messengers play a vital role in ensuring that the body responds appropriately to internal and external stimuli. Both the sympathetic and parasympathetic systems start with preganglionic neurons that release acetylcholine or ACH. This neurotransmitter binds to nicotinic cholinergic receptors, known as NACRs, located on the postganglionic neurons, initiating the next step in the signaling process. In the sympathetic division, most postganglionic neurons secrete norepinephrine, or NA. This neurotransmitter binds to adrenergic receptors on the target cells, triggering responses like increased heart rate or pupil dilation. In contrast, Postganglionic parasympathetic neurons continue to use acetylcholine, but this time it binds to muscarinic cholinergic receptors, or MECRs, on the target tissue. These interactions help promote calming effects such as slowing the heart rate, stimulating digestion, and enhancing glandular secretion. There are notable exceptions to these general patterns. For example, sympathetic cholinergic neurons, specifically postganglionic fibers that innervate sweat glands, release acetylcholine instead of norepinephrine. This exception ensures proper thermoregulation through sweat production. This figure compares the neurotransmitters and receptors used in the sympathetic and parasympathetic pathways of the autonomic nervous system. Both pathways begin with a preganglionic neuron originating in the central nervous system. In the sympathetic pathway, the preganglionic neuron releases acetylcholine, or ACH, which binds to nicotinic receptors located on the postganglionic neuron within the autonomic ganglion. The postganglionic neuron then releases norepinephrine, which binds to adrenergic receptors on the target tissue, triggering a physiological response such as increased heart rate or pupil dilation. In contrast, the parasympathetic pathway also starts with the preganglionic neuron releasing acetylcholine onto nicotinic receptors of the postganglionic neuron. However, in this case, the postganglionic neuron releases acetylcholine again, which binds to muscarinic receptors on the target tissue. This interaction promotes calming and restorative effects, like slowing the heart rate and stimulating digestion. This comparison highlights a key difference. The sympathetic division uses norepinephrine at the target site, while the parasympathetic division uses acetylcholine at both synapses, reflecting their distinct roles in either preparing the body for action or promoting relaxation. This figure illustrates the efferent divisions of the nervous system, highlighting three distinct pathways: the parasympathetic pathway, the sympathetic pathway, and the adrenal sympathetic pathway. In the parasympathetic pathway, the preganglionic neuron originates in the central nervous system and releases acetylcholine, or ACH, onto nicotinic receptors of the postganglionic neuron. The postganglionic neuron then releases ACH onto muscarinic receptors located on target tissues, such as smooth and cardiac muscles, glands, and certain adipose tissues. This pathway is responsible for conserving energy and promoting rest and digest activities. The sympathetic pathway follows a similar structure, but differs in its neurotransmitters and target responses. The preganglionic neuron releases acetylcholine onto nicotinic receptors of the postganglionic neuron. However, the postganglionic neuron releases norepinephrine, which binds to adrenergic receptors on the target tissue. This pathway stimulates fight or flight responses, preparing the body for stressful situations. The adrenal sympathetic pathway takes a unique approach. Instead of synapsing with a postganglionic neuron, the preganglionic neuron directly stimulates the adrenal medulla by releasing acetylcholine onto nicotinic receptors. In response, the adrenal medulla releases epinephrine and norepinephrine into the bloodstream. These hormones act on distant adrenergic receptors, providing a widespread and prolonged sympathetic response that enhances physical performance during emergencies. This figure illustrates how autonomic synapses function, focusing on the release of neurotransmitters from structures known as varicosities. The process begins with the preganglionic autonomic neuron, which originates in the central nervous system and extends its axon to an autonomic ganglion. Here, it synapses with a postganglionic neuron. The postganglionic neuron's axon travels toward the target tissue, in this case, smooth muscle cells, and forms multiple varicosities along its length. A varicosity is a swelling along the axon that contains vesicles filled with neurotransmitters and mitochondria to provide the energy needed for neurotransmitter release. Unlike traditional synapses, which are localized at the axon terminal, varicosities are spread out along the axon, allowing for a broader distribution of the chemical signal over the surface of the target cells. When the postganglionic neuron is activated, the varicosities release neurotransmitters into the surrounding extracellular fluid. These chemicals then diffuse to the smooth muscle cells, binding to receptors and triggering physiological responses such as contraction or relaxation. This diffuse release system ensures that a single postganglionic neuron can influence a wide area of target tissue, which is essential for coordinated and efficient regulation of autonomic functions. This figure illustrates the process of norepinephrine release and removal at a sympathetic neuroeffector junction, showcasing how the sympathetic nervous system communicates with target tissues. The sequence begins when an action potential travels down the axon and arrives at the varicosity. This electrical signal triggers the opening of voltage-gated calcium channels, allowing calcium ions to flow into the varicosity. The influx of calcium initiates exocytosis, where norepinephrine-containing vesicles fuse with the membrane and release norepinephrine into the synaptic cleft. Once released, norepinephrine diffuses across the gap and binds to adrenergic receptors on the target cell, eliciting a specific physiological response, such as increased heart rate or vasoconstriction. The receptor activation continues until norepinephrine either diffuses away or is actively removed from the synapse. Removal occurs through two main pathways. Norepinephrine can be taken back into the varicosity through reuptake mechanisms, where it can be repackaged into vesicles for future use. Alternatively, norepinephrine can be broken down by the enzyme monoamine oxidase, preventing prolonged stimulation of the target cell. This efficient cycle ensures that norepinephrine is released rapidly when needed and removed promptly to maintain precise control over sympathetic nervous system responses. Autonomic receptors on target cells come in multiple subtypes, allowing for varied and precise physiological responses. These receptors are primarily G-protein coupled receptors, which initiate complex intracellular signaling pathways when activated. In the sympathetic nervous system, the primary receptors are adrenergic receptors, which respond to the catecholamines norepinephrine and epinephrine. These receptors are divided into two major classes: alpha receptors and beta receptors. Alpha receptors are more responsive to norepinephrine than to epinephrine. Within this class, alpha receptors cause muscle contraction or secretion through exocytosis, playing a role in functions like blood vessel constriction. In contrast, alpha receptors work by decreasing levels of cyclic AMP within cells, leading to smooth muscle relaxation or reduced secretion. Beta receptors differ in their affinity for catecholamines. Beta receptors respond equally to both epinephrine and norepinephrine, influencing heart rate and contractility. Beta receptors are more sensitive to epinephrine, promoting effects like airway relaxation and vasodilation. On the other hand, beta receptors are more responsive to norepinephrine and are involved in processes such as lipolysis in adipose tissue. In the parasympathetic nervous system, target cells are stimulated by muscarinic receptors, which respond to acetylcholine and mediate functions such as slowed heart rate, increased digestion, and enhanced glandular secretion. This table presents an overview of the agonists and antagonists of various neurotransmitter receptors, highlighting how different chemicals can enhance or inhibit receptor activity. Understanding these interactions is essential in fields like pharmacology and physiology, as they directly influence how medications and toxins affect the body. Starting with cholinergic receptors, which respond to the neurotransmitter acetylcholine. The muscarinic receptors are activated by the agonist muscarine, which stimulates parasympathetic responses like increased glandular secretions. These receptors can be blocked by antagonists such as atropine and scopolamine, which are used clinically to reduce saliva production during surgery or to prevent motion sickness. Indirectly, medications known as acetylcholinesterase inhibitors, such as neostigmine, prolong the action of acetylcholine by preventing its breakdown. For nicotinic receptors, the agonist is nicotine, which stimulates these receptors, enhancing neural transmission. Antagonists include alpha-bungarotoxin, which blocks nicotinic receptors at muscle junctions, leading to paralysis, and tetraethylammonium, which blocks ganglionic receptors. Another antagonist, curare, interferes with neuromuscular transmission, causing muscle relaxation or paralysis. Moving to adrenergic receptors, which respond to the neurotransmitters norepinephrine and epinephrine. Alpha receptors are activated by the agonist phenylephrine, a drug often used to relieve nasal congestion. Antagonists, known as alpha blockers, relax blood vessels, making them useful for treating high blood pressure. Beta receptors have specific subtypes that respond differently to various chemicals. The agonists isoproterenol and albuterol activate beta receptors, with albuterol being commonly used to open airways in patients with asthma. Antagonists, known as beta blockers, include medications like propranolol and metoprolol, which reduce heart rate and lower blood pressure, making them helpful in managing heart conditions. In addition to direct agonists and antagonists, there are indirect agents that influence neurotransmitter activity. For example, substances like ephedrine and amphetamine stimulate the release of norepinephrine, while drugs like cocaine prevent the reuptake of norepinephrine, prolonging its action at the receptor site. The adrenal medulla, located at the center of the adrenal glands, plays a crucial role in the sympathetic division of the autonomic nervous system by secreting catecholamines, which include epinephrine and norepinephrine. Functionally, the adrenal medulla is considered a modified sympathetic ganglion. It is innervated by sympathetic preganglionic fibers that originate from the spinal cord. Unlike typical sympathetic pathways that use postganglionic neurons with axons to reach target organs, the adrenal medulla contains specialized chromaffin cells that serve as postganglionic neurons but lack axons. When stimulated by the preganglionic fibers, these chromaffin cells release epinephrine directly into the bloodstream. Because the hormone travels through the blood to reach distant organs, it is classified as a neurohormone. This process allows for a rapid and widespread sympathetic response, enhancing the body's ability to handle stressful situations through increased heart rate, elevated blood pressure, and energy mobilization. The adrenal medulla's ability to secrete catecholamines ensures that the fight or flight response is not only immediate, but also sustained for as long as the body needs to confront or escape danger. This figure illustrates the structure and function of the adrenal medulla, emphasizing its role in the sympathetic division of the autonomic nervous system. The adrenal glands are located on top of each kidney, as shown in the body diagram. Each adrenal gland consists of two distinct regions: the adrenal cortex, which functions as a true endocrine gland, and the adrenal medulla, which operates as a modified sympathetic ganglion. Signals from the central nervous system travel through a preganglionic sympathetic neuron, originating in the spinal cord. The preganglionic neuron releases the neurotransmitter acetylcholine onto the chromaffin cells within the adrenal medulla. These chromaffin cells act as modified postganglionic sympathetic neurons, but differ from typical neurons because they lack axons. Upon stimulation, the chromaffin cells secrete epinephrine directly into the bloodstream. Because it travels through the blood to reach distant organs and tissues, epinephrine is classified as a neurohormone rather than a traditional neurotransmitter. Once in circulation, epinephrine binds to receptors on various target tissues, triggering widespread effects associated with the fight or flight response. These effects include increased heart rate, elevated blood pressure, bronchodilation, and enhanced energy availability, all aimed at preparing the body to handle stressful situations. The adrenal medulla's ability to release epinephrine into the blood ensures a fast and long-lasting sympathetic response, complementing the rapid but more localized neural pathways of the sympathetic nervous system. This section focuses on the Somatic Motor Division, which is responsible for controlling voluntary movements of skeletal muscles. A somatic motor pathway is distinct from autonomic pathways because it consists of only one neuron. This single neuron originates in the central nervous system, either from the brain, via cranial nerves, or from the ventral horn of the spinal cord. The neuron's axon is myelinated, which allows for faster transmission of electrical signals. These axons are very long and always excitatory, meaning they stimulate the muscle to contract rather than inhibit it. Near the target muscle, the axon branches, and each terminal end innervates a single skeletal muscle fiber, ensuring precise muscle control. The connection between the motor neuron and the muscle fiber occurs at the neuromuscular junction, which is a specialized synapse. At this junction, the motor end plate on the muscle cell membrane contains nicotinic cholinergic receptors, which are receptor ion channels that respond to the neurotransmitter acetylcholine. When an action potential reaches the end of the motor neuron, acetylcholine is released into the synaptic cleft. It then binds to these receptors, triggering an influx of ions that leads to muscle contraction. To prevent continuous stimulation of the muscle, the enzyme acetylcholinesterase breaks down acetylcholine in the synaptic cleft, allowing the muscle to relax after contraction. This one neuron pathway ensures quick and direct communication between the nervous system and skeletal muscles, enabling rapid and voluntary movement. This figure illustrates the somatic motor pathway, which is a key component of the efferent divisions of the nervous system responsible for controlling voluntary movements of skeletal muscles. The pathway begins with a single motor neuron whose cell body is located in the central nervous system, either in the brain or the spinal cord. This neuron sends a myelinated axon directly to the target skeletal muscle without any synapses along the way. At the neuromuscular junction, the axon terminal releases the neurotransmitter acetylcholine into the synaptic cleft. The acetylcholine molecules then bind to nicotinic receptors located on the membrane of the skeletal muscle fiber. The activation of these receptors triggers an influx of ions into the muscle cell, initiating a chain of events that leads to muscle contraction. This direct one-neuron pathway allows for fast and precise muscle movements, enabling actions such as walking, lifting objects, and speaking. Because this pathway is always excitatory, stimulation of the motor neuron invariably leads to contraction of the muscle. After the contraction, the enzyme acetylcholinesterase breaks down the acetylcholine in the synaptic cleft, allowing the muscle to relax and prepare for the next signal. This efficient communication system ensures that voluntary movements are quick, controlled, and coordinated to meet the body's immediate demands. This figure illustrates the structure and function of somatic motor neurons and the neuromuscular junction, which is the specialized synapse between a motor neuron and a skeletal muscle fiber. In the image on the left, the somatic motor neuron originates from the central nervous system and sends its axon toward the target skeletal muscle fiber. As the axon approaches the muscle, it branches at its distal end to form multiple axon terminals. Each axon terminal interacts with a specific part of the muscle fiber known as the motor end plate. This arrangement ensures that a single motor neuron can control multiple muscle fibers, allowing for coordinated muscle contraction. Surrounding the axon are Schwann cells, which provide insulation and support for faster signal transmission. The image on the right shows a close-up view of the motor end plate. This region of the muscle membrane contains a high concentration of acetylcholine receptors, specifically nicotinic receptors, which are essential for receiving the signal from the motor neuron. When an action potential reaches the axon terminal, it triggers the release of the neurotransmitter acetylcholine into the synaptic cleft. Acetylcholine then binds to these receptors, initiating an electrical change in the muscle fiber that ultimately leads to muscle contraction. The Schwann cell sheath surrounds the axon terminal, providing structural support and helping to maintain the health of the neuron. Additionally, mitochondria within the terminal supply the energy required for the release of neurotransmitters. The neuromuscular junction is vital for voluntary movements, allowing the nervous system to communicate efficiently with skeletal muscles. This direct and rapid connection ensures that actions like walking, grasping, and speaking are performed with precision and speed. This figure provides a detailed overview of how somatic motor neurons communicate with skeletal muscle fibers at the neuromuscular junction, leading to muscle contraction. In the top left illustration, you can see the neuromuscular junction, where the axon terminal of a motor neuron forms a synapse with the motor end plate of a skeletal muscle fiber. Within the axon terminal are synaptic vesicles filled with the neurotransmitter acetylcholine. The muscle membrane at this junction contains nicotinic receptors, which are specialized to respond to acetylcholine. The top right panel explains the sequence of events leading to neurotransmitter release. An action potential traveling down the motor neuron reaches the axon terminal, causing voltage-gated calcium channels to open. Calcium ions enter the terminal, triggering the fusion of acetylcholine-containing vesicles with the presynaptic membrane. Acetylcholine is then released into the synaptic cleft, the small space between the neuron and the muscle fiber. Acetylcholine diffuses across the synaptic cleft and binds to nicotinic receptors on the motor end plate. This binding opens ion channels, allowing sodium ions to flow into the muscle cell and potassium ions to flow out. The influx of sodium ions is greater, resulting in a net positive charge entering the muscle fiber, which depolarizes the membrane and initiates a muscle action potential that leads to contraction. The bottom illustration shows this ion movement in detail. When acetylcholine binds to the receptor, the channel opens, allowing sodium ions to enter and potassium ions to exit the cell. This ion exchange creates the electrical impulse necessary to stimulate the muscle fiber. To prevent prolonged stimulation, acetylcholine is quickly broken down in the synaptic cleft by the enzyme acetylcholinesterase. This breakdown ensures that the muscle can relax after contraction and be ready for the next nerve signal. This rapid and coordinated communication between the neuron and muscle fiber allows for precise, voluntary muscle movements essential for activities like walking, speaking, and grasping objects. Let's wrap up with a quick summary of the two major divisions we explored in this chapter. First, we covered the autonomic division. This division of the nervous system operates involuntarily, regulating essential bodily functions like heart rate, digestion, and respiratory rate. It includes two key branches: the sympathetic division, which prepares the body for fight or flight responses, and the parasympathetic division, which promotes rest and digest activities. Together, they work to maintain homeostasis through opposing or cooperative control mechanisms. Second, we examine the somatic motor division. Unlike the autonomic system, this division controls voluntary movements of skeletal muscles. It consists of a single motor neuron pathway that transmits signals from the central nervous system directly to the muscles. Through the neuromuscular junction, the release of acetylcholine stimulates muscle fibers, allowing for precise and coordinated movements essential for everyday activities like walking, speaking, and lifting objects.